Chapter 16, Development, Stem Cells, and Cancer. This will be 16.1, that first section. What this is showing is a fly embryo, probably a fruit fly, and it is looking at three different genes. A red gene, a gene that has been labeled red, a gene that has been labeled blue, and a gene that has been labeled green. The other colors are where they're interacting. Red and green together and make yellow, red and blue are going to make purple, etc. So there is some pattern of how those genes are expressed. So a program of differential gene expression leads to the different cell types. So you're going to get a fertilized egg, but even though all the cells have the same DNA, they're not going to be expressed in the same way. The cells in the brain are going to make brain tissue. The cells in the muscles are going to make muscular tissue, completely different. So it depends on location and what those locations end up being into, cells to tissues to organs, organ systems, etc. So you're going from an egg and a sperm, then a zygote, and then that zygote has to divide. They have to differentiate, meaning that the cells have a specific path now, and change into the proteins, ultimately the cells and tissue types that they're going to be. So comparing a frog and a tadpole, 4 days. These pictures would be essentially 4 days apart, which is pretty crazy, from an undifferentiated circle to a very specifically laid out organism. So at that point, you're going to have differentiation, where the cells now are on a specific path. They might become muscle cells or cells similar to muscle cells, or they might become some sort of skeletal cells. It could be bone, it could be cartilage, something like that. Morphology is shape, so morphogenesis is giving that shape, and it's going to depend on how those genes are expressed, and a lot of it, at least some of it, is going to be determined before the egg and sperm ever come together, depending on the types of messenger RNA and proteins in the mom's egg cell. So what the mom's egg cell has is DNA, RNA, proteins, etc., that are going to be floating around and not evenly spread out. Some sides might have more of one, more of the other. These are called cytoplasmic determinants, and they are going to be some of the things that are going to set the stage for different areas getting different types of cells. So I will try to play this animation, and hopefully it doesn't sound like crap.
Cell-to-cell signaling, with molecules carrying signals from sending cells to receiving or target cells, is a key mechanism in the coordination of cellular activities. In most cases, a signal molecule acts by binding to a receptor protein in the plasma membrane of the target cell and activating a series of changes, a signal transduction pathway, that triggers a specific response within the cell. A signal molecule binds to a specific receptor in the plasma membrane of the target cell. This activates a series of relay proteins, which in turn activates a transcription factor. The activated transcription factor attaches to the DNA and triggers transcription of a specific gene. The mRNA message is translated into a new protein, which changes the structure or function of the receiving cell.
So there you go. It's okay. So here's an example. You've got your unfertilized egg, and let's say there's two different proteins that are going to cause different kinds of signals. And there's more green triangles on the right, more brown circles on the left. They become fertilized by a sperm. Now you've got a zygote, a fertilized egg, but they're still very uneven. As they go through their first cell division, the left side ends up with more of these browns that are going to give a certain signal, and the right side ends up with more of the greens that are going to give another signal. So they end up being induced. Those cells that start as cells that can become anything get taken on a path. Those molecules induce those cells to become something specialized. Sometimes it's a matter of one cell inducing another. So they're now we're looking at a 32 cell stage, early embryo, and depending on what molecules one cell is putting out, it may affect the next cell. If this molecules or these cells have already established that they're going to be on the head end, maybe they're sending a signal to be you're the abdomen or something like that. So once a cell has been determined, it's been set on a path to where it can only become a few things. It's not like a stem cell that can become anything. It's now a stem cell that can become a few things. Kind of same thing. It's going to be determined by different types of proteins and different messenger RNAs as far as the molecules that are going to determine which way it goes. For example, an example we'll go through are the signals within the genes that take cells that are generally anything and make them specifically become muscle cells. So here's our embryonic precursor cell. It can become anything, any kind of cell. And it's going to have the same DNA as every other cell. There's going to be a master gene and then other genes that are specific to muscles. If this is going to end up being in an eye or in bone, these are going to stay off. If chemicals take it on the path of being what's called a myoblast, blasts are making, myo means muscle. Now it's on a course. It cannot become, say, nervous tissue anymore. It's going to be a muscle. So what happens is chemicals are going to turn on this master regulatory gene, MyoD. DNA gets copied to RNA, which gets copied into amino acids, essentially proteins. So now you've got this master protein thing that has been made. What it does is it will turn on all of the other muscle genes. So now that this thing is becoming a muscle cell, this MyoD protein getting made, making more, is going to turn on another gene, and that might turn on other genes that make things for the muscle, myosin, proteins, etc. Another thing, important part of the development of any sort of embryo is not only the growth of cells, but the death of cells, which seems odd, but that's sort of helping mold the shape of that organism. So apoptosis, programmed cell death. Certain signals in the cell that make them go away and other cells survive. And that's part of development. It's also part of life. As far as when cells get old, when cells get damaged, they go through apoptosis, programmed cell death. That's not a bad thing. In fact, the lack of it is a bad thing. So the cell sort of is on self-destruct mode. It packages up its parts into sort of blobs, and then those blobs are going to get eaten up. Because if they just fell apart, all of their chemical contents, especially ones that can digest things, could break down these cells around them. So this is a, I believe, white blood cell, and on the left we have a normal white blood cell. On the right we have a white blood cell that is going through apoptosis. The stuff that is, or the things inside the cell are being put into essential little vesicles, and those vesicles are going to be eliminated one at a time so that they don't influence or damage this good cell. Hands and feet's an interesting example of this. So you've got, this is, I know it's green, but it's false color. It's a mouse. So this is early on, and you've got webbing. You start as big, blobby, fin-like things. You have tissue in between the digits, interdigital tissue, those cells are going to go through programmed cell death, and you end up with separate digits. We do this as humans. In fact, sometimes humans don't have some of those turned on, and you have people that are born with webbed feet, webbed fingers. It's possible. And then pattern and position. So deciding on an embryo, what's going to be the head end, what's going to be the tail end, what's going to be the back, what's going to be the belly. This is all going to be set up by signals, by turning on certain genes. Two model organisms we're going to look at. Drosophila, I hope I'm saying that right, Drosophila, so I'm going to have to say it a lot. Melanogaster, that is the common fruit fly. We know a ton about that. We know a ton about a nematode called C. elegans. And we can take what we've learned about those and transfer that to other things. Like before, the egg of the mother is going to determine some of the axes. What's going to be the front, what's going to be the back, depending on the proteins floating around within that egg. So anatomy and physiology stuff, the head end is anterior, the butt is posterior. Dorsal's back, ventral is belly. And then left and right are left and right. We'd have to be looking down on it to see that. So in the ovary, it's got different types of cells. Those cells are going to have different chemicals on different sides. And the egg gets fertilized. That fertile egg is going to develop into different stages and different parts of the body. So here's our developing egg. We've got our egg here, our nucleus. There is a nerve cell, which is helping it survive, as well as a follicle cell around it. They're going to have influences on that egg. As the nerve cells go away, and that egg becomes bigger and bigger and bigger, it's going to also start to develop its shell. Eventually, it's going to get fertilized by sperm. It's going to be laid, not the fly, the egg. And depending on the layout of that egg, you're going to start to lay out body segments. Something that is definitely going to become a head up here. Something's definitely going to become a tail back here. By the way, green is head, brown is thorax, and orange is the abdomen. So there you go. Now we've got a larval stage, essentially a maggot stage, will further develop into a fly. Homeotic genes or homeobox genes, or sometimes just called Hox genes, are going to be master genes that are going to help control where there's a head, where there's a tail, etc. And sometimes they go wrong, very, very wrong. Normal wild type, we've got antenna coming out here like there should be, and here's a mutant and there's legs out of its antenna. That's not good. Yep, still creepy. So some of the mutations are bad. Some mutations are good. Most of them don't do anything. Bad ones, embryotic lethals, that means that they die as an embryo. They never are born, or they never get an egg laid. 120 genes in the fruit fly just for segmentation. So maternal effect genes are going to lay out that body plan. If those genes are strange or mutant in the mother, she's going to pass those on, and they're going to affect orientation, polarity, head, tail, that kind of stuff. A specific one, bicoid, is going to be active in the front of the body. So it's going to determine where the head is. If an embryo from an egg, from a mother that doesn't have this gene, doesn't develop the front half of its body, which is as creepy as it sounds. So normal larva, you get a definite head region, you get a definite middle, you get a definite tail. And the tail region is with these sections called A6, A7, A8. If it doesn't have that bicoid, it builds a tail, A6, A7, A8 on this side, and on this side as well, A6, A7, A8. Two tails, no head. It will not survive. That's creepy. So that would be one example of a compound that is establishing how that is going to be set up. And it's going to be more on one side. So the messenger RNA that makes this protein called bicoid, it's going to be more on one side or the other. Once it gets fertilized, you go from mRNA to protein. Protein gets produced, ends up being on one end. That's going to be your head end. The end that doesn't have that is going to be the tail end. To prove this, if you take this protein or the mRNA that makes the protein and you put it into other areas that aren't the head, they will form heads.
Over the last few years, research has shown that mRNA molecules in the unfertilized egg of Drosophila determine the embryonic axes and future body plan of the fruit fly. The mRNA molecule that specifies the anterior-posterior head-tail axis of the embryo is transcribed from an egg polarity gene which codes for a protein called bicoid. This gene is active, mRNA is made, in the nurse cells that surround the developing Drosophila egg. The bicoid mRNA, once transcribed, is deposited into the Drosophila egg asymmetrically. More bicoid mRNA is deposited at one end of the egg than the other. Fertilization of the egg triggers the translation of the maternal bicoid mRNA into protein. The bicoid protein then diffuses through the egg, forming a gradient. The bicoid protein gradient establishes the polarity of the embryo along the anterior-posterior axis. The head and thorax will develop in the zones of high bicoid concentration. The abdomen will develop in the region of low bicoid concentration.
Hopefully the last few minutes my microphone hasn't been bad because I had the microphone facing the speaker. So you take that mRNA, you put it on one end, that's going to make the head. This is before, this is after, ends up being the end with the head on it. Yep. So protein helps determine pattern. It also shows that there's more controlling factors in the egg than we were aware of originally. Ooh, 18 minutes. Sorry about that.